All right, in this video, we want to talk about the lysogenic life cycle of viruses, and we'll compare this life cycle to the lytic life cycle we've previously learned about. So, the lysogenic life cycle is one of the two types of viral replication. Again, previously, we've talked about this lytic life cycle. We talked about how quick it is, how it creates a large number of viral particles, and how it results in the death of the cell in this release step. Today, let's think about this lysogenic life cycle. As we can see from this diagram, the lysogenic life cycle interacts with the lytic life cycle. So we'll see how the two life cycles interact with one another, and we'll think about the steps that make the lysogenic life cycle unique. So, lysogenic viral replication. What characteristics does it have in general? So in comparison to this lytic life cycle, which was very fast, for a bacterial virus, we talked about this lytic life cycle taking 30 minutes or so as a rough number. This lysogenic life cycle is much slower. We can't apply a time, we'll talk about why, but basically this is much slower. The virus only divides when the cell divides, and cells don't divide nearly as fast as viruses do. All right, the lysogenic life cycle also only produces one additional virus each time the host cell replicates. So again, when this bacterial cell becomes two cells, we now have two viruses. But each sort of generation here only produces one additional virus. Remember, the lytic life cycle could produce hundreds of viruses in one generation. Last, during this lysogenic life cycle, we can say that the virus is sort of hidden, or some describe it as being dormant inside the host cell. And so this can be a really tricky thing that viruses do, and it can be one of the reasons that viruses cause the types of disease or the progression of disease that we observe. And we'll talk about that later in the semester. All right, so let's think about how lysogenic viral replication happens. In my video about lytic viral replication, we talked about 5 unique steps to that life cycle. Here, we're just going to say, well, there are some more steps. I don't know if we want to count them specifically, but I'm going to add some additional terminology, and we'll talk about what that means. So during the lysogenic viral replication, we're going to go through these steps in this order. The first one is attachment. This is the exact same kind of attachment that happened in the lytic life cycle. So I've labeled it step 1 here. You also saw it as step 1 in my lytic video. After attachment, we're going to get entry or penetration. Again, this step is 100% the same in both the lytic life cycle and the lysogenic life cycle. This is where the two life cycles deviate, though. So instead of continuing with some numbered steps, I'm going to say the next step is step 2A. We're going to call it insertion, and we'll add some detail about what that means in a slide or two. After insertion, I described the lysogenic life cycle as having these replication steps. Again, we'll add some detail about what that means in just a moment. And then finally, we'll call it step 2C. We're going to refer to a step called induction. We'll see what happens to the virus during induction. After induction, or immediately following induction, is when the virus re-enters the exact same steps that were happening in the lytic life cycle. So lytic life cycle has attachment and entry. In the lytic life cycle, immediately after entry is synthesis or biosynthesis. The lysogenic life cycle has these extra steps happening between entry and synthesis. Again, synthesis is going to be just like in lytic life cycle. After that, we're going to get assembly, just like we did in the lytic life cycle. And then after that, we're going to get release, exactly the same as we had in the lytic life cycle. So let's look at these steps and think about what's happening during each of them. So first step, attachment. Again, this is the exact same as what happens in the lytic life cycle. The virus is floating around in the environment. It bumps into a potential host cell. If that host cell has the appropriate surface markers on the outside, the virus is going to stick. The reason the virus sticks is because the virus has the right spike proteins that will interact with the markers on the outside of the host cell. Sort of like a lock and a key. The spike protein's the key, the markers on the outside of the host cell are the lock. So we get a virus sticking to the outside of a bacterial cell. Again, these spike proteins are attaching to surface markers on the host. Again, that is exactly the same in the lytic life cycle and the lysogenic life cycle. Next, we've got entry. Again, during entry, the virus injects its genome into the host cell, so its DNA or RNA leaves the capsid and goes into the cytoplasm of the host cell. That's it. That's entry. That's exactly the same in both the lytic and the lysogenic life cycles. Again, this is where the lysogenic life cycle deviates and we get a unique step. So in the lysogenic life cycle, we're going to call this step 2A, we get insertion. So the top of this diagram is entry. The virus genetic material is entering the cytoplasm of the cell. Insertion is when that viral DNA becomes incorporated into the genome of the bacterial cell. So the virus genome is incorporated into the bacterial genome. Their pieces of DNA are connected together. When a virus does this, we have a special piece of terminology for the virus at this point. We call the virus at this point, when it is nothing more than DNA attached to the bacterial DNA, we call the virus a prophage. So we can see the prophage here, the viral DNA attached to this circular bacterial chromosome. Once the viral DNA is incorporated into the bacterial DNA, it stays there for some time. And we can proceed to the next step, which is I would just refer to as replication. So at this point, every time the host cell divides to make two cells, it's going to make a second copy of the viral DNA. So we have a host cell here, the top middle. We've got the bacterial DNA and the viral DNA. When this cell decides it is going to divide, it makes a second copy of its DNA. We see that happening here. And then when it's complete, we see that there are two distinct copies of its DNA inside the cell. Because the virus DNA is part of the bacterial DNA, we now have two copies of the viral DNA. This cell can then divide, becoming two different cells. And again, we now have two cells, each containing one copy of the virus. We started with one cell containing one copy of the virus. So that virus just reproduced. There are now more viruses, but we make one at a time every time the host cell divides. That will go on for an indeterminate amount of time. It depends on the virus. It depends on the conditions that the host cell is growing in. But at some point in the future, we'll progress to step 2C, induction. Induction happens when some appropriate stimulus causes the prophage to remove itself from the bacterial genome and re-enter the lytic life cycle. So, a virus that is going through the lysogenic life cycle is reproducing with the cell and is existing just as this DNA incorporated into the bacterial genome. But again, at some point, this virus gets stimulated, it becomes activated, and it re-enters the lytic life cycle at the synthesis step. So it removes itself from the DNA, and then we immediately go to synthesis. Again, synthesis is a step that's in common between both the lysogenic and lytic life cycles. The very first thing that happens during synthesis is that the virus destroys the host genome. That way the host cell cannot follow the instructions in that DNA. So we chop up the host genome, and we have the viral genome. Then the virus uses the host cellular machinery to make all the viral components. So we make a whole bunch of the pieces that we need for new viruses. After we have all the pieces for new viruses, we go through assembly, where the viral components are put together. Again, you can see in this electron micrograph that there are complete viral particles inside this cell. Once all of the viruses have been assembled inside that cell, we move on to the last step, release, where those viruses exit the host cell when the cell lyses, or goes through lysis. So basically the cell pops and it releases all those viral particles into the environment. And that's it. That's the lysogenic life cycle. Again, there's a lot of overlap with the lytic life cycle, but there are a few steps that make it unique. And specifically, the slow replication, the long sort of dormancy inside of a cell is what sets it apart from the lytic life cycle. So as always, I hope you'll let me know if you have any questions related to this video, and I look forward to talking to you all in class or on the discussion boards.